Malaria is a life-threatening disease caused by parasites that are transmitted to people through the bites of infected mosquitoes. It is preventable and curable. In 2016, there was an estimated of 216 million cases of malaria in 91 countries. The sub-Saharan Africa region carried a high share of global burden. In 2016, the region was home to 90% malaria cases and deaths. Not only malaria is a life-threatening disease in sub-Saharan Africa, it also contributes to 30 million cases in Greater Mekong sub-regions. One child died every single 2 minutes due to malaria. The most effective treatment for malaria has always been working in the patient's blood by killing the parasites. However, recently the parasite has been mutated and trying to fight back the attackers, which is the antimalarial drugs. And therefore, there is an urgent need for a novel antimalarial tablet that tackle this problem. At Franklin Pharma, we strive to carry out the most insightful science in our research field by the best researchers and best research students. Our aim is to develop and identify the novel compounds to tackle these resistant problems in these state-of-the-art laboratories.
Malarial parasites consume the hemoglobin either for the construction of its own protein and energy or to create a space inside the red blood cells. Hemoglobin is digested in the acid organelle, namely the digestive vacuole. Such process results in the production of the toxic heme molecules. In order to detoxify the heme, the parasite would crystallize it to form the hemozoin, a non-toxic molecule which accumulates in red blood cells. The mode of action of 4-aminoquinolines is still remaining unclear and require further research for further understanding of its antimalarial activity. The proposed mechanism is that it hampers the polymerization of heme, leading to an accumulation of heme inside the red blood cells. As heme is known to be toxic for the parasites, the accumulation of which results in the parasite cell lysis. Additionally, it is found that the basicity of 4-aminoquinolines contribute to its antimalarial activities due to the stability to accumulate in the digestive vacuole. In their unionized form, they are able to permeate the membrane and accumulate in the digestive vacuole, which is acidic. And here, when they are ionized inside the vacuole, they became trapped inside. So this is the pharmacophore of the 4-aminoquinoline. The chlorine atom here is responsible to inhibit the hemozoin formation. These two aromatic rings, they are responsible to form stacking interaction with the hemoglobin. And here, these two groups with nitrogen atoms, they're responsible for the acid trapping in the digestive vacuole. And for the chain here, we can add a linker just to increase activity against the resistant strains.
After doing a research on 20 potential compounds that have similar structure to chloroquine, we have identified 3 compounds that could be our lead. They are pinonaridine, terbuquine, and amodiaquine. However, we chose amodiaquine as our lead because of the 2 reasons. The first reason is because of the presence of the aromatic ring that contribute to its rigidity and the increase in accumulation in the infected red blood cell. These 2 features result in an enhanced antimalarial activity. Secondly, amodiaquine have a similar structure to terbuquine, the most potent antimalarial compound. But amodiaquine is smaller in size and less lipophilic, which means we could propose further modification to enhance its bioavailability. Having said that, amodiaquine has similar toxicity profile to terbuquine. Therefore, our goal is to reduce its toxicity profile while maintaining its potency.
We plan to make modifications in the following 3 sites in amodiaquine. One of the modifications is to add substituents at the 5th position. Substituents considered include an additional methyl group which increases the activity against chloroquine-resistant species and parallodynyl methyl group for reducing liver toxicity. The rest of our modifications aim to increase the activity of our compounds against chloroquine-resistant strains. Modifications include replacing the hydroxyl group at the 4th position to classical isoster such as fluoro or amino group, making an interchange of side chains between the 3rd and the 4th position, and switching the terminal amine from diethyl group to pyrrolidinol or tertiary butyl group.
A maximum of 4 synthetic substances are all we need to come up with 24 solutions to malaria. 18 of the solutions will be bioavailable and have excellent ability working against chloroquine-resistant strains. The remaining 6 compounds are backup candidates which are less bioavailable, yet they have even better ability against drug resistance. All our compounds can be produced efficiently within a short period of time, because only simple agents such as ethanol and hydrochloric acid are required in our synthesis, and starting materials such as different terminal amines are currently available from our suppliers. Therefore, once we started, all compounds will be ready within a maximum of 2 weeks.
We're going to show you how to produce one of our products, compound 14, as an example of synthesis. This is starting material 1A obtained from our suppliers. A pyrrolidinol amine is added to starting material 1A via nucleophilic aromatic substitution to form intermediate 5D. The protecting group of intermediate 5D is removed by hydrolysis of amide to form intermediate 10D. Finally, aminoquinoline rings are added to intermediate 10D via nucleophilic aromatic substitution and resulted in our product, compound 14.
After synthesizing target compounds, it is crucial to carry out different biological screenings to assess their biological activities. In this project, in vitro hypoxanthine and in vivo assays are the proposed choices because they are the gold standards for antimalarial screening recommended by the WHO. Hypoxanthine assay measure the viability of parasites after treatment with different concentration of drugs. Parasites incorporate purine into nucleic acid for growth. This assay utilizes this characteristic to determine the degree of radiolabeled hypoxanthine incorporation in incubated parasites. IC50 values of target compounds can be obtained after these measurements. Hypoxanthine assay has high sensitivity and reproducibility, therefore it is applicable for high throughput screening, which allows screening large number of compounds at one time. Following the in vitro assay, in vivo assay, the 4-day suppression test will be carried out. Mice are treated with parasites before they are actually treated with drugs. After that, blood smear test is carried out. The percentage of parasitemia and the ED value for our potential compound can be calculated. The use of in vivo assay ensures our effective compounds possess an acceptable balance between pharmacokinetic and pharmacodynamic properties. Hence, the risk of attrition is reduced during clinical development. Consequently, this minimizes the expenditure for resources between beginning and the failure of our projects. In vivo assay allows those compounds that are successful in mouse model to be subsequently tested in primate models prior to phase 1 clinical trials in man.
No toxic metabolites are formed following metabolism phase 1 and 2. The majority of our target compounds are expected to be metabolized through deamination and oxidation, whilst others are directly went through glucuronidation and glutathione conjugation. A minority of our target compounds are excreted directly as parent compounds in the urine or conjugated with glutamine before being excreted. For example, target compound number 2 undergoes P450-mediated oxidation to form a quinolamine intermediate. Formation of this intermediate is only possible because there is a 1:4 relationship between the amino and phenolic groups of the aromatic substituent. Glutathione then conjugates with the intermediate to give a non-toxic metabolite, which is then excreted.
This is the Gantt chart outlining the timeline for our project. We propose that our project can start in February next year after receiving the commercially available starting materials and reagents. 3 medicinal chemists will be in charge to identify appropriate synthetic pathways in 3 weeks. Moving on to the synthesis and purification of target compounds, 3 medicinal chemists and 1 lab assistant will be recruited to carry out this task within 3 months. Then they will prepare biological assays to assess our target compounds. In vitro assay will require 5 days, and in vivo assay will require about 2 months. 4 medicinal chemists and 2 biologists will carry out data analysis and write up the final proposal in 3 months. Quality assurance will be carried out from the beginning of our project. In total, 6 months will be required to complete the project.
Franklin Pharma for the future of no malaria.